Okay, everyone, this is the online lecture for Chapter 10 on photosynthesis. I've been having some technical issues with these, and I found that I'm going to have to limit each of these to about a half hour because of file size limitation issues. So I'm not going to get this one done in one presentation here. It's probably going to be split up into two, so part one, part two, all right? Okay, so this chapter is on photosynthesis, as I said, all right? And we know that photosynthesis is the process that converts solar energy into chemical energy. By chemical energy, I mean the energy in the covalent bonds between carbon, hydrogen, and oxygen atoms in a sugar molecule. So, either directly or indirectly, photosynthesis nourishes almost the entire living world. It is the process that produces the food that you eat, whether it's plant or animal. Think about it, plant is a direct result of photosynthesis. Animal meat is an indirect result. The animal eats plant, right? The cow eats plant, you come along, eat the cow, so you're getting that photosynthetic product indirectly through the cow. Okay, so let's define a few things. First of all right, autotrophs. Autotrophs are referred to as self-feeders. They are any organism that is capable of producing its own food or feeding itself without consuming some other organism or some other part of some other organism. So plants are autotrophs. They combine carbon dioxide with water in the presence of sunlight, and they convert that CO2 and water to sugar molecules, which becomes their food. Autotrophs are referred to as the producers of the environment because precisely that, they produce food for everything else. They build organic molecules, sugars primarily, from inorganic molecules like CO2 and water. More specifically, plants are what we call photoautotrophs, meaning they use sunlight as a means of autotrophic food production. Okay, there are some types of organisms called chemoautotrophs that use chemicals to produce organic molecules, but they're rare, not that common, and we're not going to focus on them here. Okay, so take a look at this tree here. That green canopy of leaves that you see above that tree, that's a complex solar collecting array, essentially. That's what a leaf is. This is a solar collecting structure. All right, so the chlorophyll, which gives those leaves the green color, is the chemical component of the chloroplast, the pigment, rather, that absorbs sunlight and traps the energy of that sunlight and ultimately makes it available to the process that builds the sugar molecule. All right, this is important for the plant because this is how the plant's feeding itself, but you can also see it's important for other organisms as well, like the squirrel that's feeding on a plant product, in this case, what looks like an acorn. Okay, so again, either directly or indirectly, photosynthesis is feeding the entire world. Okay, so photosynthesis occurs in plants, algae, certain single-celled eukaryotes, protists primarily, and also in some prokaryotes. So these organisms are feeding themselves, but they also feed everything else on the planet. Here you see some examples. So we have plants, for example, in a rainforest. You could also have, a picture there of plants in a temperate forest or a grassland or whatever, a farm field. We have multicellular algae. Algae are not plants, they're a different group of organisms. This is a picture of kelp, which are a type of brown algae. We have single-celled eukaryotes. Actually, these are euglena, and you can see the individual chloroplasts inside those cells. All right, we have a group of bacteria, prokaryotes, called cyanobacteria that are capable of running photosynthesis. We saw a few examples of those in our cell structure lab, if you remember looking at Gliocapsa and Oscillatoria. And then we have a fairly unique type of bacteria called purple sulfur bacteria. They run a different form of photosynthesis from what we're going to talk about in this chapter. They're not that common, but it is a different way to run photosynthesis to make sugars. All right, another definition. Heterotrophs. Heterotrophs are other feeders. These are organisms that cannot produce their own food directly like an autotroph can, So they have to get their food from other organisms by consuming those other organisms or parts of those organisms or whatever. Animals, for example, are heterotrophs. We are heterotrophs. We have to get our food by consuming other organisms, either plant or animal. So the heterotrophs depend on the photoautotrophs both for food, primarily as sugar, and also oxygen. Photosynthesis produces oxygen as a byproduct, and we saw in the last chapter how it is that we use oxygen. Oxygen sits at the bottom of our transport chains and pulls the electrons down, allowing us to produce a large amount of ATP. All right, so plants are obviously very important for us. Give us our food. They also give us the oxygen we need to break down that food and produce ATP. Okay, here you see an example of a heterotroph, right? That little guy there eating that acorn. He can't stand the sunlight and make his own food, so he has to actually consume a product of a plant. Okay, and the same way that we do. So through the process then of photosynthesis and also cellular respiration, carbon is actually cycled between inorganic and organic forms in the environment, and this process is called the carbon cycle. All the carbon that we have on the planet is all the carbon that has ever been on the planet and that ever will be on the planet. So it's just constantly being recycled, and there are carbon atoms on this planet that are billions of years old, and it's extremely likely, in fact, a certainty, that there are carbon atoms in you right now that used to be in other people in the past, or that used to be in a dinosaur 80 million years ago, or whatever, or in possibly even the first eukaryotic cell 2.6 billion years ago, because all these carbons are constantly being recycled, and that's what the carbon cycle is all about. And so I have a video animation here from the publisher of the textbook to talk about the carbon cycle. So let me play this, and then we'll come back and move on with the chapter.
Could a carbon atom that was once part of Albert Einstein be in you? All living things need to take in carbon-containing molecules to fuel their activity and build their cells. This is true for busy students and for all organisms. Let's look at how carbon atoms move through the carbon cycle in the Arctic tundra. Carbon atoms from carbon dioxide are incorporated into living things by producers, such as this plant. Carbon dioxide enters a leaf, moves into a cell, and goes into a chloroplast. There, in the process of photosynthesis, carbon dioxide is used to make sugar molecules. The sugars can be used to build larger carbon-containing molecules, carbohydrates, lipids, proteins, and nucleic acids. In this way, carbon that was in the air is now part of the plant. When a plant is eaten by an herbivore, or primary consumer, carbon in plant tissue enters the primary consumer's digestive system. There, plant molecules are broken down. These small molecules are then used by the primary consumer to make larger molecules for building its own tissues. In this way, some of the carbon that was in a plant becomes carbon in a primary consumer. Similarly, when a primary consumer is eaten by a higher-level consumer, some of the carbon in the primary consumer is incorporated into the higher-level consumer. Carbon-containing molecules in dead plant material, animal feces, and dead organisms are taken up by decomposers, such as fungi and bacteria. Decomposers break down these materials and incorporate some of the carbon atoms into their own bodies. We have now seen how carbon moves from carbon dioxide to producers, to primary consumers, to higher-level consumers, and to decomposers. How does carbon get back to the atmosphere? During cellular respiration, carbon-containing molecules are broken down, and ATP is made. Carbon dioxide is released as a byproduct and returned to the atmosphere by producers, consumers, and decomposers. Overall, this return of carbon dioxide by cellular respiration is closely balanced by its removal from the atmosphere through photosynthesis. The global carbon cycle involves living things on land and in rivers, lakes, and oceans. And, by the way, considering the huge number of carbon atoms in each person and how quickly carbon atoms move through the carbon cycle, there is almost certainly a little of Einstein in each of us.
Okay, so now let's move on to talking about a little bit about plant structure, plant anatomy, as it relates to photosynthesis. So we know that leaves are the major sites of photosynthetic activity. Chloroplasts are found mainly in a certain layer or cell layer inside the leaf. That cell layer is called the mesophyll. Layer, the mesophyll layer. So mesophyll cells typically contain maybe 30 to 40 chloroplasts. The number varies, but that's probably a pretty good estimate for an average. And so these cells are primarily responsible for running photosynthesis. Now, one thing about photosynthesis, okay, is that it does require a source of CO2 and it produces oxygen. And so there has to be a way for the CO2 to enter the leaf and get into the cells. There has to be a way for the oxygen that's produced as a byproduct to get out, because if O2 builds up, it can become toxic. It can actually begin to inhibit photosynthesis and shut down the process. So there are microscopic openings on the underside of the leaf called stomata, and they allow for what's called gas exchange. So this is CO2 in, so CO2 in and O2 out. All right. All right. Now, also, as far as the chloroplast goes, we talked about chloroplast structure a little bit. Let me change colors here a little bit back in, I think it was chapter 6. And so a chloroplast is sort of a disc-shaped structure like this. It has an outer membrane, and also that's lined by an inner membrane. Both of those membranes are smooth. There's a fluid inside called the stroma, and part of the reaction of photosynthesis happens in that fluid. But then we also have a third set of membranes inside that space that is looped and folded in such a way that it looks like a stack of dinner plates, plates stacked on top of one another. So each of these structures is called a granum. All right, and each little disc inside that structure is called a thylakoid. All right, so it's the thylakoid membrane that you see there, looped and folded that way, that contains the chlorophyll. And so again, we see an example here of looping and folding, and the reason for that looping and folding is to increase surface area, as we've seen. That usually means that that membrane is important, and this membrane, the thylakoid membrane, is certainly important because it contains the chlorophyll. The more chlorophyll the chloroplast has, the more sugar ultimately can be produced. All right, let's take a look. So here we have, get rid of that. Okay, here we have an example of leaf anatomy. So if you look at the mesophyll leaf here in cross section, what you can see is the mesophyll layer right there. Okay, so each of those cells then inside is responsible for running photosynthesis. There is a layer of cells on both sides of the leaf. Those cells really don't participate much in photosynthesis. They're more or less involved in waterproofing the leaf and helping the leaf to prevent being dried out. Here you can see stomata, microscopic openings allowing for gas exchange, as we talked about. The plant can actually control those. It can open them up, close them down, depending what it needs to do. If we zoom in onto an individual cell, now you can see all the individual chloroplasts inside of that. Okay. All right. Now, let me change colors here again. All right. Okay, so now you see over here an actual chloroplast. You can see the outer membrane that's smooth, the inner membrane that's smooth, and all that. You can see the third set of membranes, the thylakoids. You can see a granum there, one shown in cross section here, And this way you can see it's actually a looped and folded membrane. It's not like we actually have individual plates. Again, those plate-like structures are called thylakoids. You see the term right there. But inside you see it's actually open space, looping and folding a membrane. And again, that's just to increase surface area so that more chlorophyll can be packed inside that chloroplast. All right, this is below here, this is an electron micrograph view of a single chloroplast. You can see a granum there, one there, there, and so on. Again, the fluid is called the stroma, and that's where part of the reaction of photosynthesis actually takes place. All right, so what is our summary equation then for photosynthesis? So our plant takes 6 molecules of carbon dioxide, combines those with 12 molecules of water, and in the presence of light energy, it rearranges those atoms into C6H12O6 plus 6 molecules of O2 plus 6 molecules of water. All right, this is our glucose molecule. That's our sugar. Okay, so this process is anabolic because it's building CO2 and water into a sugar. It is therefore also endergonic, as we learned in Chapter 8. It is also therefore non-spontaneous. It will not move forward on its own without an input of energy. Okay, so, and if you look at it, it's effectively the reverse cell respiration. If you run the reaction that way, combine glucose with water, you break it down to glucose with oxygen, you break it down to water and CO2, that's cell respiration. And so cell respiration and photosynthesis are exactly the reverse of each other, okay? The important point here is that, I spent a good amount of time in Chapter 9 talking about how you should think of cell respiration as a mining process. It mines high energy electrons out of our food and uses their energy to build ATP. Well, you might have wondered, maybe, how those high energy electrons get into the food in the first place. Photosynthesis does it. Where does the energy come from to energize those electrons and then pack them into a food molecule? Sunlight. All right. So let's look at the source, or actually where it is, the destination of all these different atoms that we see in the reactants. Okay, so like we said, we have 6 molecules of CO2. All of those carbons in CO2 go into glucose. If glucose has 6 carbons, CO2 has 1 carbon, you need 6 copies of CO2 to get enough carbons to build glucose. All right, we have 6 times 2, 12 oxygens in CO2, and 6 of them end up in our glucose molecule. The other 6 end up in our water molecule. Okay, what about the water that we get as have as a reactant? Well, there are 12 times 2, sorry about that. Let me go back. 12 times 2, 24 hydrogens, and fully 12 of them end up in our sugar, and the other 12 end up back in other water molecules. The oxygen from that water goes off into the atmosphere, and that's what we breathe. So think of photosynthesis as a process that splits water molecules apart and liberates the oxygen to the atmosphere. So why is photosynthesis splitting water apart? Water is what provides the electrons. All the electrons we mine out of our food through cell respiration to use it to produce ATP, those electrons ultimately came from water. Photosynthesis pulls them out of water, energizes them with sunlight, packs them into a sugar molecule. That cell respiration then mines back out of the sugar molecule and uses to build ATP. So this is how photosynthesis and cell respiration are linked. Okay, so like cell respiration, photosynthesis is a redox process. Remember, oil rig. Oxidation is loss of electrons, reduction is gain. Okay, so let's look at our equation again for photosynthesis. Again, we have 6 CO2 plus 6 H2O gives C6H12O6 plus 6 O2. All right, so what is being oxidized here and what is being reduced? Well, first of all, think of this as a process of carbon fixation. So remember, fixation means to convert something from inorganic to organic form. So these inorganic carbons that you see in CO2 are converted to carbon in organic form. That's fixation, all right? So whenever you fix something, you have to add hydrogen and electrons to it. You have to reduce it. So that carbon in CO2 is being reduced to carbon as glucose. That is the fixation step. It's carbon going from inorganic form as CO2 to organic form as glucose. All right? Well, if you have reduction, you also have to have oxidation. So what's being oxidized? Well, I said in the previous slide that water is being split and electrons are pulled out of it. If something loses electrons, it's being oxidized, correct? So the water is being oxidized to oxygen. That's the oxidation step. That's where the electrons are coming out, okay? And water goes off into the atmosphere and we breathe that. All right. So overall, photosynthesis consists of two main parts, and you see both of those parts right here. We have, first of all, what's called the light reactions. That happens in the thylakoid membrane where the chlorophyll is. And secondly, we have what's called the Calvin cycle, okay? The Calvin cycle is what actually builds the sugar molecule. Okay, so what do the light reactions do? Well, the light reactions do a number of things. First of all, as we've already said, they split water, and that provides a source of electrons to funnel into the process. Secondly, this process releases oxygen, which is good for us because we need it. Third, light reactions reduce an electron carrier molecule called NADP plus to NADPH. So you'll recall in cell respiration we had two electron carrier molecules. I called them electron buckets. We had NAD plus being reduced to NADH. We had FAD being reduced to FADH2. And those are just two different molecules that carried electrons to electron transport chains. Well, we don't have those two molecules here. Instead, we have NADP plus, NADPH. All right. Fourth thing that light reactions do is they produce ATP through a process called photophosphorylation. Make it fit. There we go. Photophosphorylation. Okay. So you need a supply of ATP because the Calvin cycle is anabolic and endergonic, and it's going to build sugar. So what's going on in the light reactions is that we have, there we go. We have an input of water. Okay, water comes in, it provides the electrons. We have light coming in as the energy source. We have an input of NADP plus and ADP plus phosphate. They both come in, and the energy and the electrons from water are used to build ATP and NADPH. We get oxygen out as a byproduct. Okay, the ATP and the NADPH then are funneled into the Calvin cycle, which uses the energy of ATP and the electrons and hydrogen of NADPH as building material to combine with CO2 and build a sugar. This is important. The CO2 only provides carbon and oxygen. Well, you also need hydrogen to build a sugar, and that hydrogen comes from the NADPH. Okay, it was loaded into NADP plus along with the electrons that always follow protons, as you recall. And then thus we get a sugar molecule out. Okay, so again, this process then, the Calvin cycle in particular, is the carbon fixation step, converting inorganic carbon as CO2 to organic carbon as sugar. Okay, so think of chloroplasts as solar-powered chemical factories. The thylakoids transform light energy into the chemical energy of ATP and NADPH. All right. If we're going to talk about that and understand really what's going on there in all these processes of photosynthesis, it helps to understand something about light, the nature of light, or sunlight in particular. So light is a form of electromagnetic electromagnetic energy, okay, or electromagnetic radiation if you want to call it that, all right? Now, there are many different forms of electromagnetic energy, as we'll see here in a little bit, and all of them travel in waves. So think of light as traveling in rhythmic waves, sort of like a wave moving across the surface of a lake or the ocean, okay? Now, we have different forms of electromagnetic energy, and we can define each of them by what's called wavelength, which is the distance between two peaks or the distance between two troughs. That's wavelength. Different forms of electromagnetic radiation, such as UV, infrared, visible light, for example, have different wavelengths that characterize them. Now, here's the rule. Shorter wavelengths mean higher energy radiation. Longer wavelengths mean lower energy radiation. All right, we're going to see later on, it also can be helpful to think of light as a particle, okay, which we call the photon. We'll see that more in a minute. So in some aspects of photosynthesis, it's helpful to think of light as a wave. In others, it's helpful to think of it as a particle. It actually does exist in both forms. Depending on how you measure it, it can both appear to be a wave and a particle. That's a characteristic that Einstein talked about of light. He called the duality or dual nature of light. Light's a weird thing. Okay, so we have what's called the electromagnetic spectrum, which shows us the wide range of wavelengths that electromagnetic energy comes in and the different forms of energy that results in, okay? And as I mentioned a minute ago, sometimes we can think of light as a particle called a photon. When we get to the light reactions, we'll talk about light as a photon from then on. All right, this is the electromagnetic spectrum. And so what you see is everything from gamma rays way over here on the left all the way over to radio waves on the right. Gamma rays are very high energy because they have extremely small wavelengths. That is 10 to the minus fifth nanometers. So a nanometer is 1/billionth of a meter. 10 to the minus fifth is 1/10,000th. So gamma rays are 1/10,000th of 1/billionth of a meter in wavelength. Extremely small wavelengths. And they're extremely high energy. They are extremely dangerous. X-rays to the right, a little bit larger wavelength, but still very dangerous. UV, a little bit larger wavelength still, but still can be dangerous. High energy UV can cause mistakes or errors, problems with your DNA if you're laying on a beach, for example, without sunscreen on, and that's the basis of skin cancer. Here in the middle, we have this little sliver of light called the visible spectrum. That's the light that our eye can actually see, and it's also true of most organisms, although some can actually see in the UV on the left and infrared on the right. Humans can't. On the right, we have infrared. To the right of that, we have microwaves, then we have radio waves. Wavelengths getting longer as we go to the right. Notice in radio waves, we have wavelengths of 1,000 meters. You're talking about a kilometer. So we have radio waves, going through our skulls all day long from all of our electronic equipment, radio towers, cell phone towers, but it doesn't hurt us because the wavelengths are so long, the energy is so low. Let's go back to visible light. So here we have the visible spectrum. So maybe you remember from elementary school, Roy G. Biv, right? Roy G. Biv tells you the colors from right to left. So we have red on the far right, orange, yellow, green, blue, indigo, violet. So we have Roy G. Biv going from right to left. And so our shortest wavelengths are 380, that's the purple and violet. Longest is the red, around 750, that's the red. Okay. And it turns out that not all of these wavelengths of light are needed or responsible for running photosynthesis. Turns out it's mostly the purple and the violet on this end, purple-violet, blue to some extent as well, and the red, orange. The green plays no role in photosynthesis, and this is actually why we see plants as green, because plants just get rid of that light back to the environment. They don't absorb it. Chlorophyll doesn't absorb it. That's why when you look at a plant, it appears green, because they're not absorbing the green light, they're absorbing the other colors that you therefore can't see. Okay, so pigments are substances that absorb visible light. Different pigments absorb different wavelengths, and the wavelengths that are not absorbed are either reflected off, let's say, the leaf surface, or they're transmitted entirely through the leaf or the pigment, okay? So leaves appear green, as I said, because chlorophyll reflects and transmits green light, and that's what you see here. So here we have a chloroplast, and you have visible light coming in, which contains all the colors of the rainbow. And what happens is that as it impacts those thylakoid membranes with the chlorophyll green light is either reflected or green light is transmitted. The other colors primarily are absorbed, and so it's the energy of those colors that's mostly responsible for running photosynthesis. Okay, so we have a machine called a spectrophotometer that's very useful for understanding these sorts of things. Maybe if you've had a chemistry lab already, you've used a spectrophotometer to analyze concentration of solutes in a solution or something like that. A spectrophotometer measures the pigment's the pigment's ability to absorb various wavelengths. So it sends a light beam through a pigment, like chlorophyll, and measures the fraction of light transmitted to or at each wavelength. Okay, here's the idea. So, by the way, all of this that you see here is happening inside the spectrophotometer machine, okay? So we have a light source, we have a prism that separates that visible light into the colors of the spectrum. There's a plate with an opening in it that allows only some of the light to come through. So for example, you see that the machine now is set to allow only green wavelengths of light to come into our sample. We have a chlorophyll sample. We know how much green light's going in. We can measure through a photoelectric tube how much is coming out. And because chlorophyll rather, doesn't absorb green light, you can see we have very high transmittance, almost 100%. That means low absorption. Most of what's going in is also coming out. That's transmittance. Now, what if we were to set the machine to allow only blue wavelengths of light to come through? Well, chlorophyll absorbs blue quite a bit, so you have a lot of blue light coming in, but very little coming out. That is low transmittance, right? Near 0%, high absorption, okay? All right, so I think that's gonna be a good place to stop. I've hit my sort of half-hour mark here, so I'm gonna go ahead and stop, and I'll pick up with the next slide in part 2 of this, okay? All right.